Next generation sequencing technologies are the modern day sequencing technologies. If we look into the history of sequencing technologies, we see that Human Genome Project was sequenced by using Sanger sequencing methods. Sanger sequencing method has been there for about 25 years till we reach in year 2000 when there was a company called as Lynx Therapeutics, which was later on named as Solexa, that marketed massively parallel signature sequencing technique in year 2000. This technique led to the development and discovery of next generation sequencing machines. For the last time, I'm sure guys, so we are looking into this evolution of sequencing technology with a time scale. We have seen that significant milestones if we look into it. So we have seen that DNA was discovered in 1953. First sequence of 24 base pairs was published by Maxim and Gilbert in 73. Sanger sequencing came in 1977. Obviously both of them, Sanger and Gilbert, they shared a Nobel Prize in 1980 for these discoveries. Another distinction is development of PCR. And then we see that automated sequencers, they came into being in 1987. The first of them that was presented by ABI, Applied Biosystems. It was called as Prism 373 machine. Capillary sequencers, they came into the picture again by ABI in 1996. Then we see here comes the next generation sequencing systems. So first of them that was there in year 2005, that was the GS20 system, 454 technology. We will look into these technologies in detail later on. And Solexa presented their genome analyzer in 2006, whereas the Applied Biosystem, ABI, gave its machine called as Solid, that came in 2007. And then we have seen there is a single molecule sequencers. Basically it was what we nowadays called as the third generation, so we don't need to amplify them. In the next generation sequencing, we amplify the sequences with the help of the PCR, and this PCR got some technical issues, so they were kind of bypassed in these technologies here. Then ion torrent, which is again next generation sequencing machine, came in 2011. We see the Pacific Biosciences also introduced a PAS biosystem in 2011, same year. So again it's a single molecule sequencer. And Oxford Nanopore Technologies, they have demonstrated the use of their ultra long single molecule reads, which came in 2012. So let's see what next generation sequencing is. So next generation sequencing is a catch-all term that can also be known as high-throughput sequencing or massively parallel signature sequencing. So what happens here is that these techniques are used by different companies, and they have developed their different systems. So all of them, we put them together as the next generation sequencers. For example, we have Illumina sequencing, we have Roche 454, we have ion torrent and solid sequencing. So all of them, they are put into the NGS or next generation sequencing technologies. What they do, so NGS machines, they sequence they perform massively parallel sequencing, where they sequence millions of the fragments all together, and there is a high throughput. So the throughput means high processing. So we can take the sequences, we break them into short fragments, and we put them together and we sequence them. So normally there are millions of the sequences which are put together and sequenced together, and at the same time we can have more than 500,000 sequencing reactions running in parallel in these machines. Here, for instance, we can see an Illumina flow cell. Normally these sequences, they are sequenced in those lanes. So we see here there are 8 different lanes. So this is actually a glass slide like thing. So our sequences, they come and flow through them, and then within these lines they pass on. And if we take the cross section of these lines, we see what we call them as these stations. So in these stations we have the primers which are attached. So individual fragments, they come and attach to these primers, and then they are sequenced over here. So in this way we can have parallel sequencing of millions of these sequences all together at the same time. So as a whole, this speeds up the process, but at the same time we get a huge amount of data. The advantages of this system is obviously we don't need to have clean rooms for that, and obviously the whole sequencing reaction is there in this slide like thing. In the NGS technologies, we can have different generations of those sequencers. If we look into the Sanger sequencing, we can say it was first generation, whereas the second or the next generation is what we are talking about today, the NGS machines. And in the third generation, which we have right here, we'll talk about that later, so in which the amplification through the PCR has been eliminated, and the molecules, DNA molecules, are directly sequenced. Different technologies which use that technique, third generation, or we can also call it as next to next generation sequencing. These are Helicose, PassBio, Ion Torrent, and Oxford Nanopore. Here are some of the sequencers, the next generation sequencers. We have this GS Flex 454 from Roche. We have HiSeq machine, which is very frequently used from Illumina. We have ABI Solid here, we have an Ion Torrent, and we also have a GS Junior machine over here. Illumina has been mostly used because of its popularity and because of its flow design techniques in which we are using those flow cells, and there is another technique which is the Polyny PCR. Rest of the machines, they are using the emulsion PCR, so they have some issues with them, so it got more success. Advantages of this next generation sequencers is that they produce, they can sequence DNA or RNA molecules in a quick time, and obviously as compared to the other, if we use the Sanger sequencing, that is slow, and that can also be expensive in terms of having so many gels. So it bypasses all those steps, and there is the direct sequencing, and which is very efficient. So in parallel you have so many reactions all together, and that saves your time. But obviously there is an issue that you have your read sizes are really small. Here is a general workflow for next generation sequencing. We take our material, DNA or RNA, and then we convert them into the fragments that may be processed into our machines. We call this step as library preparation, and after library preparation we do that PCR, we amplify those fragments. So actually what we want here is we want to have so many same molecule fragments. So in this way, when we do the sequencing, we will have our strong signals. So there are two types of PCRs, one is emulsion PCR and one is Polyny. So emulsion has been used in Ion Torrent, 454, and Solid. Solid can also make use of Polyny PCR, but Polyny PCR is mainly exhibited in Illumina machines. So we conclude that NGS Technologies, they sequence millions of sequences altogether, and they sequence DNA in relatively less amount of money and in quick frame of time.